Good morning. It's April 15th, and this is your daily brief in science. Here's everything you need to know. A new study reveals alarming insights into the cancer risks associated with CT scans, a diagnostic tool widely used across the United States. Led by Dr. Rebecca Smith-Bindman from the University of California, San Francisco, and published in JAMA Internal Medicine, the research estimates that around 93 million CT scans were performed in the U.S. in 2023, impacting 61.5 million individuals. The study predicts that these scans could account for approximately 5% of all new cancer cases each year. Among the types of cancer projected to arise from radiation exposure are lung cancer, colon cancer, leukemia, and bladder cancer, with CT scans of the abdomen and pelvis contributing significantly to these cases. Particularly concerning is the heightened risk faced by children and infants, especially those under 1 year of age, who have a tenfold increased risk from head scans. The research highlights considerable variability in radiation doses from CT scans, with some patients receiving excessive exposure. This has led to calls for better management of radiation risks, advocating for the minimization of unnecessary scans and the optimization of radiation doses to reduce potential cancer risks. While CT scans play a crucial role in diagnosing various medical conditions, including cancer, their use has surged by 30% since 2007, raising questions about the balance between their benefits and associated risks. Previous studies have focused primarily on atomic bomb survivors, but recent findings indicate a link between CT scan radiation exposure and increased cancer rates in children and young adults. The study emphasizes the need for ongoing research to further explore the long-term effects of low-dose ionizing radiation from medical imaging. Co-author Malini Mahendra stresses the importance of informing families about the cancer risks related to pediatric CT scans and calls for improved communication from healthcare providers. However, some experts, such as Mark Supanik from Rush University, have raised concerns about the accuracy of the risk estimates, suggesting they may be overestimated based on the assumptions used in the analysis. Additionally, the study's reliance on data from Japanese atomic bomb survivors may not accurately reflect the risks for the contemporary U.S. population. A recent study has unveiled remarkable insights into the communication abilities of wild bonobos, challenging long-held beliefs about the uniqueness of human language. Conducted by researchers from the University of Zurich and Harvard University, the study took place in the Kokolapori Community Reserve in the Democratic Republic of Congo. The research found that bonobos frequently combine their calls in ways that suggest complex meaning relationships. This characteristic was previously thought to be exclusive to human language, raising intriguing questions about the evolution of communication. The implications of this study suggest that the roots of language's compositional nature may extend back to a common ancestor shared by humans and bonobos, dating back approximately 7 to 13 million years. Compositionality, a central feature of language, allows phrases to derive meaning from both individual elements and their arrangement. In the context of bonobo communication, compositionality can manifest in trivial ways, where meaning is simply the sum of its parts, or in non-trivial ways, where one call modifies the meaning of another, similar to human language. To further this understanding, researchers created a bonobo dictionary, cataloging various bonobo calls and their meanings. This significant advancement enhances our comprehension of animal communication and emphasizes the sophisticated vocal abilities of bonobos, challenging the notion that complex vocal combinations are a human-exclusive trait. A major advancement in neuroscience has emerged with the creation of the largest functional map of a brain to date. This groundbreaking study utilized a genetically modified mouse that watched clips from popular films like The Matrix and Star Wars. The project is part of the MI-CRONS initiative and involved over 150 scientists from various U.S. institutions. It was published in the journal Nature on April 9, 2025. Researchers have likened this foundational work to the Human Genome Project, expressing hope that it will lead to significant breakthroughs in understanding and treating brain diseases. The connectomy revealed that neurons activating together can form connections over longer distances, offering new insights into the brain's complex wiring. Findings also indicated that inhibitory neurons can target specific distant cells rather than just nearby ones, suggesting a more intricate network of neural inhibition. The research traced communication between neurons through 500 million junctions known as synapses, providing valuable insights into brain function. The team recorded activity from 76,000 neurons in the occipital lobe, which is critical for visual processing, and examined the anatomical features of these brain cells with an electron microscope. To reconstruct the neural connections in 3D, researchers sliced the brain tissue into over 25,000 thin layers, capturing nearly 100 million high-resolution images. Artificial intelligence played a crucial role in tracing the wiring of the neurons, allowing for individual identification of connections. The total wiring was estimated to exceed 5 kilometers in length. The machine learning algorithm mapped 200,000 cells and 523 million synaptic connections, making this connectomy 3 times larger than that of a human brain segment. The dataset is publicly available for global scientific research, potentially enhancing understanding of neurological disorders such as Alzheimer's and multiple sclerosis. However, despite its size, the dataset remains incomplete, with some neurons missing and orphan extensions detected, indicating the need for further manual verification and refinement. A recent study from NYU Langone Health has unveiled a new type of immune cell that may hold the key to preventing food allergies. These cells, known as tolerogenic dendritic cells, play a crucial role in immune regulation, particularly against food allergies. According to Dr. Dan Littman, the senior author of the study, ROR gamma T expressing dendritic cells are essential for maintaining this balance within the immune system. Published in the journal Nature on April 14, 2025, the research confirmed the existence of human equivalents of these dendritic cells through detailed analysis of human intestinal tissue and sequencing data. While the study has established the presence of these cells, further research is needed to understand their abundance and roles in immune tolerance beyond the intestines. Experiments conducted on mice revealed that those lacking functional tolerogenic dendritic cells were significantly more susceptible to food allergies and asthma. This finding emphasizes the protective role these cells play, as the absence of tolerogenic dendritic cells resulted in fewer regulatory T cells and an increase in inflammatory T cells, raising allergy risk. The study also identified that these specialized dendritic cells require specific proteins, including retinoic acid-related orphan receptor gamma T, or ROR gamma T, and PR domain containing 16, or PRDM16, to function properly. The implications of these findings suggest potential future applications in developing innovative treatments for food allergies by leveraging these dendritic cells to enhance regulatory T cell responses, ultimately allowing the immune system to tolerate food proteins effectively. An international research team has made strides in solving a long-standing mystery about carbon-rich asteroids and their potential role in the origin of life. This breakthrough is detailed in a study published in Nature Astronomy, which analyzed nearly 8,500 meteoroids and meteorite impacts using data from 19 fireball observation networks across 39 countries. Despite their abundance in space, carbon-rich asteroids account for less than 5% of the meteorites found on Earth. The researchers found that Earth's atmosphere and the Sun act as significant filters, destroying fragile carbonaceous meteoroids before they can reach the surface. Dr. Hadrienne de Villepoix pointed out that many of these meteoroids break apart due to heating as they near the Sun, while those that do survive are more likely to endure atmospheric entry. The study emphasizes that meteoroids generated by tidal disruptions are particularly fragile and seldom survive the journey through the atmosphere. Carbon-rich meteorites are crucial as they contain essential ingredients for life, including water and organic molecules. The findings from this research could influence future asteroid missions, impact hazard assessments, and theories regarding the delivery of water and organic compounds to Earth. Dr. Patrick Schober noted that this research may change scientists' understanding of meteorite collections and the origins of life on Earth. The limited number of carbon-rich meteorites in current collections poses a risk of an incomplete understanding of space materials, highlighting the importance of this study. Other contributing institutions include the Astronomical Institute of the Romanian Academy, the National Museum of National History, and Aix-Marseille University, with support from the International Center for Radio Astronomy Research. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.